For more than 5 centuries, nobody knew with certainty where Richard III's body had gone. The last English king to die in battle had disappeared into history almost as completely as his grave. Then, in 2012, archaeologists opened the ground beneath a Leicester car park. What they found was a skeleton buried exactly where historical records suggested Richard III's lost church had once stood. The bones belonged to a man who had died at roughly the right age. His skeleton showed severe scoliosis. His body carried injuries consistent with violent combat. And then came the DNA. One genetic line matched living relatives connected through Richard's mother. But another genetic line did something far more complicated. It did not match the male relatives who were supposed to share Richard's paternal lineage. The discovery did not simply identify a medieval king. It revealed that somewhere inside a royal family tree stretching across centuries, the biological story and the historical record had separated. And that strange contradiction is part of what makes the identification of Richard III so remarkable. Richard III died on August 22, 1485, at the Battle of Bosworth. His defeat ended his short reign and opened the way for Henry Tudor, who became Henry VII. According to historical accounts, Richard's body was brought back to Leicester and buried in the church of the Franciscan Grey Friars. But the friary eventually disappeared. The grave was lost. Centuries passed. Buildings changed. The medieval landscape vanished beneath the modern city. By the 20th century, the exact location of Richard's burial had become a historical mystery. The search eventually focused on the former Grey Friars site in Leicester. In August 2012, the University of Leicester, working with the Richard III Society and Leicester City Council, began excavations. The project was looking for a lost royal grave. Instead, archaeologists found something much more useful. A skeleton. And almost immediately, several details began to align. The skeleton was male. The individual appeared to have been between 30 and 34 years old when he died. Richard III was 32. The remains also showed severe scoliosis, a curvature of the spine that would have made one shoulder higher than the other. That detail mattered because contemporary descriptions of Richard included an asymmetry of the shoulders. The skeleton also carried multiple injuries associated with the circumstances of death. And the burial itself was unusual. The individual had been placed in the choir area of the former Grey Friars Church, precisely the kind of location expected for Richard III's burial. Radiocarbon dating and stable isotope analysis also placed the remains within the appropriate historical period and indicated a high-status diet. None of these facts alone could prove identity. A skeleton does not come with a medieval name tag. But taken together, they created a remarkably specific profile. The question was no longer simply whether the bones could belong to Richard III. The question became whether science could connect those bones to Richard's surviving family tree. This created an unusual problem. Richard III left no known living descendants. So researchers could not simply compare the skeleton's DNA with a direct child or grandchild. Instead, Professor Turrey-King and her colleagues had to look for relatives connected through specific lines of descent. That distinction is critical. Most of your DNA is inherited through a complicated mixture of ancestors. After many generations, the amount of DNA shared with a distant relative can become difficult to use for a simple identification. But two parts of human inheritance offer unusually clear pathways through deep family history. Mitochondrial DNA passes from mothers to their children. The Y chromosome passes along the direct male line from father to son. Those two genetic paths gave researchers something they could follow across centuries. The mitochondrial line became the first major breakthrough. Researchers identified living people who descended through the female line from Richard's mother, Cecily Neville. Among them was Michael Ibsen. When the mitochondrial DNA from Richard's suspected remains was compared with Ibsen's, the result was a perfect whole mitochondrial genome match. A second living female line relative, Wendy Duldig, differed by only a single base. That was consistent with the genealogical relationship and mutation expected across the generations. The genetic evidence was no longer pointing vaguely toward the Plantagenet family. It was pointing directly into Richard III's maternal line. But mitochondrial DNA was only one side of the investigation. The male line produced a complication. Richard III genealogy could be traced through the Plantagenet male line to Edward III and then through John of Gaunt to male line descendants of the Somerset family. Researchers tested 5 living male line relatives for showed Y chromosome results consistent with the expected family connection. One did not, and the Y chromosome from the skeleton did not match the living male line relatives. At first glance, that sounds like a problem serious enough to destroy the identification. But genetics does not work like a simple family tree drawn on paper. A Y chromosome follows biological fatherhood, not legal or historical fatherhood. If a child is fathered by a man other than the man recorded as the father, the Y chromosome chain can break. Researchers refer to this as a false paternity event. Historical estimates used in the study suggested such events can occur at roughly 1 to 2% per generation. Across 19 generations, even a relatively low rate creates a real possibility of a break somewhere in the chain. That meant the Y chromosome mismatch did not automatically mean the skeleton was not Richard III. Instead, it created a second mystery. The maternal genetic line pointed toward Richard. The archaeological evidence pointed toward Richard. The physical evidence pointed toward Richard. But the paternal genetic line had apparently broken somewhere along the family tree. And that raised a much larger historical possibility. At least one biological father in the documented Plantagenet Somerset chain may not have been the man history recorded as the father. The researchers could not determine exactly where that break occurred. They could not use the mismatch to name an individual. But the contradiction itself was real. And importantly, it did not erase the rest of the evidence. This is where the Richard III identification becomes more interesting than a simple DNA match. DNA was not used in isolation. The investigators combined genetics with archaeology, osteology, radiocarbon dating, genealogy, burial location, and historical evidence. Think of it like a forensic case in which every independent clue has to point toward the same individual. The location matched. The time period matched. The age matched. The sex matched. The spinal condition matched a historical description. The injuries were compatible with a violent death recorded at Bosworth. The burial location matched the expected location of Richard's lost grave. The mitochondrial DNA matched living relatives from his maternal line. And the genetic evidence could be combined statistically with the non-genetic evidence. The 2014 study published in Nature Communications reported a conservative overall likelihood ratio of 6.7 million in favor of the skeleton being Richard III. The researchers concluded that the evidence was overwhelming. That conclusion is important because it shows what the DNA actually did. It did not magically identify a random medieval skeleton. It became the genetic component of a much larger identification process. The evidence converged. There was another detail hidden inside the DNA analysis. Researchers were also able to predict aspects of Richard's appearance from his genetic data. The predicted hair and eye color were consistent with one early portrait of Richard III. That did not prove that the portrait was an exact photographic representation. But it provided another piece of compatibility between the genetic evidence and the surviving historical imagery. And this changes how the famous discovery should be understood. The story is often reduced to one dramatic sentence. Scientists found Richard III DNA. But that is not what happened. They found an unidentified skeleton. They developed a historical and archaeological case. They reconstructed a family network. They identified living relatives whose inheritance patterns could provide meaningful comparisons. They recovered ancient DNA. They compared maternal and paternal genetic lines. They encountered a contradiction. Then they combined all the evidence using statistical analysis. The identity emerged from convergence. That distinction matters. Because DNA can be extraordinarily powerful, but ancient DNA does not automatically answer every historical question. In this case, the strongest conclusion came from several independent lines of evidence agreeing with one another, and one line disagreeing in a way that genetics itself could explain. By the middle of the investigation, the story had transformed. This was no longer simply a search for a lost king. It had become a test of whether modern genetics could reconstruct a 500-year-old identity. And the answer was yes. If this kind of historical DNA investigation is worth following, subscribing helps keep these discoveries coming. Because the most revealing part of Richard III genome was not simply that it helped identify him. It was what the genetic evidence exposed about the limits of historical family trees. For centuries, genealogies were constructed from documents, births, marriages, deaths, royal records, family histories. But those records described social and legal relationships. DNA describes biological inheritance. Most of the time, those two stories overlap. Sometimes they do not. Richard III's Y chromosome appears to show exactly that kind of break somewhere in the long chain connecting him with later male line relatives. And because the break could have occurred at several points over many generations, the DNA cannot tell us which historical individual was responsible. It can tell us that the biological chain and the documented chain were not identical. That is a very different claim from saying the entire royal genealogy was false. It was not. The evidence shows a specific genetic discontinuity somewhere along the relevant male line. And that uncertainty is part of the scientific conclusion, not a weakness to hide. There is another reason the discovery was so powerful. Before 2012, Richard III existed partly as a historical argument. He was king. He was defeated at Bosworth. He became the last English monarch killed in battle. His reputation was shaped by centuries of historical writing and later popular culture. But his physical remains had vanished. Then a skeleton emerged from beneath a modern city. And suddenly, the historical record could be tested against the body itself. The bones showed scoliosis. They showed injuries. The burial location fit. The dating fit. The genetics fit. Even the broad genetic prediction of appearance was compatible with surviving imagery. The king who had disappeared into a lost grave had become a subject of modern forensic science. In 2013, the University of Leicester publicly announced that the remains were Richard III. In 2015, more than 5 centuries after his death, the remains were reburied at Leicester Cathedral. The interval between his death and reburial was roughly 530 years. The lost king had been returned to a known place. But the DNA had left behind something more complicated than a solved mystery. It had shown that identity can survive even when graves disappear. It had shown that a family tree preserved in documents can carry a biological gap invisible to historians. And it had shown that ancient DNA works best not as a magic answer, but as one piece of evidence placed into a much larger investigation. Richard III's skeleton did not speak. His grave did not identify him. His bones could only tell scientists so much. But when archaeology, history, genealogy, and genetics were placed beside one another, the clues began to converge. The maternal DNA pointed toward the king. The battlefield injuries pointed toward the king. The age and burial pointed toward the king. The location pointed toward the king. And even the contradiction in the paternal line became part of the evidence rather than the end of the case. That is why the discovery beneath a Leicester car park became more than an archaeological find. It became a demonstration of how modern science can reopen a historical question that had remained unresolved for 5 centuries. The grave was lost. The genealogy was imperfect. The DNA was damaged by time. Yet the identity survived. Not in a single clue. In the convergence of many. And perhaps that is the most fitting final lesson from Richard III. History can lose a king, but it cannot necessarily erase the biological traces he left behind.